Assalamu alaikum. We will continue cytology, and this is the second part in it. Okay, it is actually a continuation of the first chapter, but we divided it into two lectures. Okay, we have reached up to Golgi apparatus that form either secretory vesicles or lysosomes. Lysosome is the next organelle. It is considered the digestive apparatus of the cell. Okay, by definition, membranous organelle containing hydrolytic enzyme considered as the digestive apparatus. It degrades nutrient into small molecule. It eliminates or degrades foreign or dead materials. Origin, yes, if you remember, RER form its proteins, which is hydrolytic enzymes. Then it will be released as transfer vesicle, received by Golgi apparatus to concentrate it and packing it to form primary lysosome. New lysosome released from Golgi apparatus. When this lysosome starts to fuse with any structure that can be digested by it, for example food particles, it will be termed as secondary lysosome. So primary lysosome, the new lysosome that haven't entered in any digestive process. Once it starts digestion, it is known as secondary lysosome. Site, yes, we can expect it is present in phagocytic cells like macrophage. A cell large in size, its function is to phagocytose foreign particles and bacteria. So it needs many lysosomes inside it to digest them. By LM, we cannot demonstrate it by H&E, which is a routine stain, or the other special stains used previously. But we can use a test known as histochemical test. A type of test and also the stain known as histochemical stain. By this test we can detect the presence of certain enzymes inside this lysosome inside the tissue. If this reaction is completed and the stain and the reagent changes the color of the tissue and stained by it, this indicates the presence of lysosome. For example, acid phosphatase is an enzyme present inside certain lysosome. If we do a reagent that needs this enzyme, okay, and this reagent is completed and changes the color of tissue, we can say now this tissue contains lysosome containing acid phosphatase. By EM, yes, by EM we can see different types, different shapes of lysosome according to its types. If it is a primary lysosome, or if it engulfs or eats certain substance inside the cell, and in this case it is known as secondary lysosome. So another way, EM can be considered as the same title of types of lysosome. We have two types: primary lysosome, which is newly released, not entering digestive process, and secondary lysosome, which means this primary lysosome fused with something to be digested, and in this case it will appear as if it is two something fused together, so it is heterogeneous in shape. Primary, just released from Golgi apparatus, appears as homogeneous vesicle like that, surrounded by a single membrane. While secondary, if you remember, I want just you to memorize this photo. Something large structures that enter the cell enter by a process known as endocytosis. If the particle is solid, we will have a phagosome, and if it is a fluid, we will have pinocytic vesicle. And large particles also can leave the cell to the outside by a process known as exocytosis. Lysosome released newly from Golgi apparatus is known as primary lysosome. This primary lysosome can fuse with something enter the cell by a process of phagocytosis, which is solid particles. Okay, it will fuse with a solid particle engulfed by phagocytosis, and in this case it will appear heterogeneous in shape and known as heterolysosome. Okay, if it fuses with a fluid, so it will fuse with pinocytic vesicle, and in this case we have multivesicular body, because the second lysosome is known as multivesicular body. It can also fuse with all the organelle inside the cell, for example all the organelle like mitochondria, and in this case known as autolysosome. Okay, the end product of digestion of any of these structures can be eliminated by the cell to the outside by exocytosis, but sometimes it retained inside the lysosome, and these known as residual parts. Okay, function of lysosomes. I didn't repeat the description of the previous data show because we have summarized it in one photo. Okay, function of lysosome. It is considered the digestive apparatus of the cell. It digests nutrient, bacteria, virus, can be considered as a defensive system of the body. It digests all the organelles. By this way, it maintains the health of the cell. Digests the whole dead cell after death, and is known as post-mortem autolysis. Then we will have the other component of the cell, which is the nucleus. Okay, we have finished the cytoplasm, the organelles, mainly membranous organelles, and now we will have the nucleus. Nucleus, by definition, is the largest and most important component of the cell. All cells in our body are nucleated except RBCs, which are red blood corpuscles of blood, and the platelets. Okay, nucleus varies according to its number, position inside the cell, and its shape. Okay, want to see nucleus by light microscope. If you remember, nuclei contain DNA and RNA, so it appears basophilic structure, either pale stained, and in this case we can term it vesicular nucleus or open-faced nucleus, like that of nerve cells, and this indicates that it is highly active nucleus. Or can appear dark stained, like that, also basophilic, and in this case we describe it as condensed, closed face nucleus. For example, small lymphocytes in our structure of the nucleus. It is formed, if you remember, from nuclear membrane, has pores or foramens like that. Inside we have chromatin material, which is the DNA mainly, okay, and we have nucleolus, and all are suspended in a soap-like structure known as nuclear sac, like cytoplasm of the cell. Starting by nuclear membrane, it appears basophilic. It formed of two membranes fused at interval to form nuclear pores. Okay, these pores allow the passage of rRNA, which are formed here by the nucleolus, to leave the nucleus and pass to the rough endoplasmic reticulum to be attached to its surface and to start protein synthesis. Also, we have inside the nucleus this chromatin. Chromatin represents the genetic material inside the cell, formed mainly of DNA attached to certain proteins like histones and non-histones, which are certain types of proteins, and by LM being DNA, so it appears basophilic. This chromatin by EM is either appears heterochromatin or euchromatin or achromatin. This heterochromatin means it is condensed chromatin. It appears dense like that. So this nucleus contains heterochromatin. It is condensed. It represents inactive chromatin, which carries inside it inactive genes, and these present inside inactive cells. And these cells appears by LM as condensed or dark stained nucleus or closed face nucleus, with a photo of EM picture of heterochromatin. The other type is the euchromatin. This means that the DNA inside the chromatin is not coiled like that here. It is extended. It is like a thread, and it is highly active. It forms a copy, duplicate, and form a copies of it. It is transformed into genes, and genes are transformed into proteins. So it is extended. It represents active genes. It is present inside cells, active in protein synthesis. Okay, and this one is known as open-face nucleus, and this one by LM appear as pale stained nucleus or vesicular nucleus, if you remember. Again, by LM, nuclei are either pale stained or vesicular with prominent nucleolus, or condensed or darkly stained nucleus. By EM, it appears either this one will appear A chromatin and this one will appear as heterochromatin. Other component inside the nucleus is the nucleolus. This is the cell. This is the cytoplasm. This is the nucleus, and this is the chromatin inside it, and this is the nucleolus. Okay. Nucleolus also appear basophilic because it contains DNA and RNA. It is the site of formation of ribosomal rRNA. It will leave through the foramen nuclear pores to reach the cytoplasm to attach to the outer surface of rough. The last component is the soap-like structure, which is nuclear sap. It is a solution, fills the spacing between the other components. It contains some enzymes and some lipoproteins. Function of the nucleus: it is the most important component of the cell. It carries genetic information. It is responsible for RNA formation for protein synthesis. It controls cell division and cell function. Now you will have the practical part related to cytology. Yes, EM or LM. Once we have seen a picture which is like all the movie black and white, it is EM, and we have to write it. This is an EM picture of, yes, a chromatic nucleus. Pale chromatin if you see like that. And this is the prominent nucleus. EM picture of heterochromatic nucleus. All these are inactive chromatin, appears dense like that. This is the trilamellar structure of two cell membranes. This is the one cell membrane, another one. Each one has trilamellae, 3 lamellae above each other, 2 electron dense and 1 electrolucent in between. This is an EM of mitochondria with its characteristic crest. Another mitochondria with crest. Yes, these are parallel tubule with ribosome attached to it, so this is EM picture of RER. We have an irregular arrangement of the smooth surface, so this is EM picture of smooth endoplasmic reticulum. This is LM or EM? Yes, LM, because it is colored. It only appears blue in colors, right? Okay, this means that these structures are basophilic. These are basophilic cell, and this is the nucleus. Inside close to the nucleus, we have unstained area. So this is the negative Golgi image inside plasma cells. Yes, these are photos stained by brown color, appears brown. This is the color of the silver stain. It is a special stain that you have mentioned. This is the LM picture of Golgi. If you remember, Golgi can be stained by silver stain. It appears as fibrils or granules, either above the nucleus or around the nucleus. Here we have a cell, appear columnar in shape or like a column or rectangle. These are the nuclei, and these are the Golgi apparatus above it. So this is an LM picture of Golgi apparatus in secretory cells stained with AG. We have rounded cell, okay, with active nucleus. These rounded cells are the nerve cell, okay. So, and inside this nerve cell we have fibril stained brown in color around the nucleus. So this is LM picture of Golgi apparatus in nerve cells stained by silver. This is an EM, not LM, right? Yes, and these are the saccules of Golgi apparatus, 3 to 10 saccules above each other. And these are the vesicles that enter, and these are the vesicles that leave, right? TV and SV. So this is an EM picture of Golgi apparatus. In all data show, you have to mention only these commands. You are not requested to see any details, to say any details. Yes, this is a vesicle, okay, appear homogeneous. This another one, but it appear heterogeneous, dark part and light part. So this is an EM picture of primary lysosome and secondary lysosome. Primary lysosome and secondary lysosomes. Yes, this is an old organelle with crest. Yes, excellent, it is mitochondria, engulfed by a vesicle. So this is an EM picture of autolysosome. Near the end. Thank you.